Hello again, biology students. This is lecture 10.2, part 2 on cell division. I'm going to just cruise through the first part of the notes by way of review, so nothing to write here. You've written this down already. We don't have a Google Doc for you for these notes, but there is a printable PDF. So there's a 10.1, 10.2 notes handout that is printable if you have a printer at home. If you don't have a printer at home, you can just write the notes out by hand. Even if you do have a printer at home, you're going to need a separate piece of paper for handwritten notes for this. So if you printed up the PDF and you're taking the notes in that form, then you are still going to need a separate piece of paper. Because these notes, you're going to take a picture and turn it in with a selfie. There's no typing up these notes. We want you to write these notes out by hand, and then your submission to us as the assignment is not just a picture of the notes, but a picture of the notes with you, a selfie of your beautiful smiling face. All right, so just by way of review, we're covering chapter 10, cell growth and division. There's our big idea. Cells undergo cell division to produce new cells, and this is a highly regulated cycle. Today we're going to look at the details of that cycle. Why do cells divide? How does a cell divide to make a new cell? We've already covered why cells divide. They need to stay small. One, it reduces the demands on the DNA, so it reduces information overload, and we know that smaller cells are more efficient because they have that larger surface area to volume ratio. So I go through this quickly because we've covered this already. Again, before a cell divides, you need 2 complete sets of DNA because each new cell needs a complete set of DNA. We know from chapter 12 that we double the DNA, so we have 2 sets of DNA. When we divide the cell, we divide the DNA, so each new cell gets a complete set of DNA. Each cell also stays small enough to maintain that high efficiency. Just a couple of images of cells dividing. 2 forms of sexual and asexual reproduction. Asexual reproduction, genetically identical offspring, fast, efficient. Disadvantages, limits to genetic variability, which means they're less adaptable to changes in the environment. That changes genetically from generation to generation is the fuel for evolution. And again, it occurs in both single and multi-celled individuals. Just some images. Again, we've covered this already. Bacteria, freshwater, hydra, and a fern plant. Sexual reproduction involves 2 parents. We know that you have tremendous genetic variability because half the DNA comes from mom, half the DNA comes from dad. Disadvantages, biologically, it's costly, it's very time consuming, and again, occurs in both single and multi-celled individuals. And again, just some images with respect to sexual reproduction. And then we learned about the structure of a chromosome. So a chromosome is a really long string of DNA organized in a really particular fashion and bundled up. We know that prokaryotes, this comes from chapter 20, do have DNA. It is that double helix, but it's a double helix that loops back on itself. Technically not considered a chromosome. Eukaryotes have chromosomes of varying numbers. The only one we really need to know is 46 in human beings. And again, just pointing out that the number itself is almost meaningless. It's really what does the DNA code for that matters. What these numbers don't tell us is how big are the chromosomes. You could have 25, we could have 26 or 32 really large chromosomes, or say 94 really small ones. And again, here's the image of our chromosome, just so we understand the structure. Here's the double helix wrapped around these proteins. That forms a nucleosome. Then we coil that, so it looks like an old school phone cord. And then we coil that, and then we loop the coiled double helix that folds back on itself. Pretty amazing, intense, efficient packaging. Again, this is how you get 6 feet of DNA in the single nucleus of a single human cell. All right, so on to the new stuff. So here's where we're going to slow down a little bit because here's where you want to actually start picking up the writing. Again, if you printed the PDF, fill the PDF in. If you have not printed the PDF, taking notes by hand. And you're really going to want 2 sheets. If we were doing this in class, we would have 2 handouts. We would have the printed PDF, and then we would have another assignment or another piece of paper that had a format on it that was holding it landscape. All right, so what is the cell cycle? A cell, the life of a cell includes both cell growth and activity, and then the actual time where the cell prepares and then divides into two respective daughter cells. We know from chapter 20, so this term is a review, the process of binary fission. Binary meaning two, fission meaning to split. That's the division of a prokaryotic cell into two genetically identical offspring cells. This is a form of asexual reproduction. We saw this image in chapter 20. I do want you to draw this, so just take a minute and pause it if you need to. But I want to say before you pause it, don't just draw a circle here because the picture is a little deceptive. I want you to actually draw like a double helix. Draw this twisted ladder, but draw a twisted ladder that loops back on itself. And then fill in these parts. Here's the cell membrane. Fill in the DNA. Again, you've got two loops of DNA. Here the cell starts to divide, and then here we have two cells, each with a loop of DNA, this loop genetically identical to that loop. Again, if you need to pause for a minute to fill this in, please do so. This is an actual micrograph of bacteria that are dividing. The background that you see here is the medium on which the bacteria were growing. I don't know if the bacteria are actually this color or if they've been stained, but you can see they're just at the edge of being done replicating. Pretty incredible image when you consider how small they are. All right, now we're going to focus on the eukaryotic cell cycle. A lot of details in this. So the eukaryotic cell cycle, and again, it's a cycle, so we're gonna end where we start. It's really two main parts. We have what's called interphase. This is a cell spends almost its entire life in interphase. This is where a cell is growing. This is where the DNA is replicated, and this is where a cell does regular cell activity. If you are a liver cell, you're spending most of your life in interphase metabolizing food. If you are a nerve cell, you're in interphase conducting electrical impulses. If you are a heart cell or a muscle cell, you're involved in facilitating movement. Most of a cell's life is in interphase. How long that is could be anywhere from weeks to months to years. Most of a cell's life is in interphase. Then we have what's called the M phase. And the M phase is the time where we actually take the cell and it's going through the process of dividing into two. So the cycle is interphase, M phase, interphase, M phase, interphase, M phase. M phase being relatively short compared to interphase. Right. M phase is further divided into what we call mitosis and then cytokinesis. And then mitosis is further divided into 4 phases. We'll get into those details in just a second. Excuse me. All right, so there's M phase divided into mitosis and cytokinesis. And we'll see some graphics here in a little bit that'll help make this clear. Don't draw this down. This isn't a very good graphic. We're going to skip this. This is where I want you to draw. If you have the PDF, you're going to need a separate piece of paper. If you're drawing this by hand, or if you're taking notes by hand and you're not using the PDF, you can put this right in with your notes. Excuse me. However, when you draw this, we're going to have to put some labels in here. Draw this really big. Have this, there's no points for conservation of space. Have this be, at least a quarter to a third of the size of your paper. All right, so I'm just going to click here. All right, so we're starting here. Here we have a single cell at this point right here. As you can see, this whole section here is called interphase. Interphase is further divided up into G1, S, and G2. We're not going to test you on the names of these subsections within interphase, but you do need to know that during interphase, these 3 things happen. Cell growth and activity. Here is where we have the DNA replicated. So we now have 2 sets of DNA, and then this is the preparation for when cells begin to divide. The G stands for gap. gap 1, the S stands for synthesis, and the G stands for gap 2. Sort of misnomers. A long time ago, when the cell cycle was first discovered, it was obvious that there was a whole lot going on in the cell here, but that there wasn't necessarily going on. There didn't seem to be a lot going on in here, hence the term gap. We now know that that's not the case. There's actually a whole lot of activity that goes on during these phases. We're not going to get into those details of what it is. We're just going to call it normal cell activity. All right, so again, we have interphase, and then we're going to move on to M phase. So draw this circle nice and big. Yes. Draw the pie chart. Yes. Label these 3 sections. Yes. Put this arrow in here. Again, if you need to pause, please do so and go ahead. Don't need to draw this picture, but I do want you to reference this. Underneath, again, if you're drawing the PDF, if you're writing on the PDF, you've got space to take these notes on the PDF, and you're drawing the cell cycle, the circle cell cycle, on a separate piece of paper. If you're just writing your notes by freehand, you can just put these next set of notes underneath your cell cycle circle, and we're just going to go back and forth between the two. All right, so here's the basic premise. Again, don't worry about these labels. Just take a look at the basic picture, what's going on here. Here's the nucleus. Here's the DNA. We've condensed, the DNA is condensed down into chromosomes. Chromosomes line up along the middle. They're connected to these little protein ropes. Remember, each chromosome is double-sided, so each half of each chromosome is identical to each other. And then these little protein ropes start getting shorter and shorter and shorter, and we literally physically pull apart the double-sided chromosomes. This stretch of DNA is identical to this stretch of DNA. This stretch of DNA is identical to this stretch of DNA. This stretch of DNA is identical to this stretch, and this stretch is identical to this stretch. So here we have 4 individual chromosomes. Here we have 4 individual chromosomes, because each of these are identical, each of these are identical. Same thing here and here. The DNA that ends up in this nucleus is identical to the DNA that ends up in this nucleus. All right, again, that's just an overview. Let's look at the details. First section is called prophase. Pro meaning first phase. The main thing that happens during this section is the copied DNA condenses down into our double-sided chromosomes. Think back to interphase. I said most of a cell's life is in interphase. During interphase, the DNA is not condensed down into the chromosomes. During most of a cell's life, the cell needs access to the DNA in its long string form. So most of the time, in the nucleus, the DNA is unwound in a form that's referred to as chromatin. It's only during that G2, gap 2 phase of interphase where all that long stringy DNA condenses down into chromosomes, right? So during that time, the DNA condenses down into the DNA prepares, and then it's during prophase that long string of DNA condenses into the visible double-sided chromosomes. Again, if you are writing the PDF, go to your separate piece of paper. If you are writing the notes by freehand, reference back up to this and label this section right here prophase. All right, so again, we have interphase, M phase, interphase, M phase. M phase is further divided into mitosis and cytokinesis. We'll apply those labels later. Here we're looking at mitosis. The first, and then mitosis is divided into 4 sections, and that first section is titled prophase. The DNA condenses and becomes visible. All right, what happens during the second stage? All right, so again, here's a cartoon image of prophase. Metaphase. Meta here means middle. The chromosomes line up along the middle of the dividing cell. All right, so here the chromosomes are lined up. This can be a little misleading. Students look at something like this and they think the chromosomes are lined up end to end to end because we're looking at a flat 2-dimensional picture. I want you to think of this picture right here more as it's a sphere, and we've cut through the middle of it. So really what we really have is a disc, right? So We have a disc, or what's sometimes referred to as the metaphase plate. We have a plate here, and the chromosomes are lined up all over the plate. They're lined up in the middle, but they're lined up in the middle all over the plate. Now, what also is going on is on this end, we have these two little yellow cellular tow trucks, and we've got another pair down here, and there's this spider web of protein ropes coming out, connecting to the chromosomes. Right, so here is our double-sided chromosomes. Here are these protein ropes that are connected to our double-sided chromosomes. This, of course, is metaphase. There's the label reference back to the graphic where you'd label metaphase. So we have prophase, metaphase. Again, remember, we're going clockwise in our circle. Prophase, metaphase. The next one is anaphase. And ana means away or apart. Each of the identical halves of each chromosome move to opposite ends of the cell. Right, so now in this case, what happens is we have these protein ropes that are connected to the chromosomes. They start to literally shorten up. They shorten up on this end, and they shorten up on this end. And as they shorten up, they pull apart those double-sided chromosomes. Now here's where we have this goofy genetics or chromosome math. Right now, we're going to call these 4 chromosomes, 4 double-sided chromosomes. But the second they split apart, we now have 8 chromosomes. So now this chromosome is identical to this one's identical to this one, this one's identical to this one, this one's identical to this one. These two used to be connected back here in the same way that these two used to be connected back here. All right, so now we have these protein ropes pulling the chromosomes apart. Anaphase. If it helps you remember, think A for away or A apart. And then lastly, the last stage of mitosis is referred to as telophase. And here, the chromosomes reach the opposite ends of the pole. The nucleus starts to reform. So what we didn't mention in the notes up here, but what happens during prophase is the nucleus starts to dissolve. Remember, eukaryotic cells, the DNA is in the nucleus. If we want to take the DNA and split it from one end to the cell and the other half to the other end of the cell, we have to remove the nucleus. So the nucleus, the cell takes apart the nucleus over here, and here the nucleus is rebuilt. Right? And then what starts to happen is the cell starts to pinch in. You can see the cell membrane starts to pinch in. Right? So now we're starting to see the formation of 2 new cells. This section is referred to as telophase, and this whole section is referred to as mitosis. Right? So we've got our divisions of the cell cycle. We have interphase and then M phase. Interphase, M phase. M phase is divided up into mitosis and cytokinesis. And that's what this last section is. This last section is called cytokinesis. Now again, I know in my graphic, I have the definition of cytokinesis above the circle. If you have the space, then squeeze it in there. Otherwise, you can put it underneath. If you're doing the freehand notes, you can put it underneath the prophase, metaphase, anaphase, telophase definitions. And then here is telophase right there. And then this whole section is called M phase. Now again, to be clear, a common misconception is that students want to include cytokinesis in mitosis. Strictly speaking, mitosis is the division of the DNA, and that's it. Mitosis is the division of the nuclear material, the DNA. Cytokinesis is the division of everything else. All those other cell parts that we learned way back in chapter 7 in the first part of biology. The cytoplasm, the ribosomes, chloroplasts if you're a plant, mitochondria, lysosomes, endoplasmic reticulum, Golgi bodies, all those cell parts, all of that stuff gets divided during cytoplasm, or during cytokinesis. Right, so interphase, M phase, interphase, M phase. M phase is divided up into mitosis and cytokinesis. Mitosis is divided up into prophase, metaphase, anaphase, telophase with those respective details. Cytokinesis is the division of the cytoplasm and everything else. I have a hard line here to make a distinction between mitosis and cytokinesis, but this line is blurry. If I'm just going to back up here for just a second, because I just want to point something out. I want to go back to the image right here. All right. So in this image right here, telophase is wrapping up here, and cytokinesis has started. So telophase is not quite done, and so my point is that telophase and cytokinesis overlap. Telophase is just about done, and then cytokinesis starts. So there's our mitosis, there's our cytokinesis, there is our M phase. So there is our cell cycle. Here is a picture of I believe it's actually a frog embryo. So it's almost 4 cells old. Here you can see the first division was this way. The next division was this way. And then the third division will be at a right angle to that plane, and it'll be this way. And then we'll have 8 cells. And then each of those 8 cells will in turn divide this way, and then that way. Very early on in development, this process is incredibly fast. At your age, that rate has slowed down, and it depends upon the cell. Some cells go through mitosis faster than others. For instance, skin cells. The skin that you are in today, you will literally be in a new set of skin 30 days from now. Your skin gets replaced that quickly. Your bone cells takes much longer. You get a new skeleton like every 6 or 7 years, something like that. Of course, it's not replaced overnight, right? It's that gradual replacement. Some cells die, new cells come in. Liver cells last about 6 months, something like that. A red blood cell lasts about 30 days. So depending upon the cell, the life of a cell varies. So the life of interphase varies. Mitosis can be anywhere between 6 to 10 hours, maybe a bit longer depending upon the cell. So relatively short compared to the regular life of a cell. All right, so that's it. I hope that was helpful. Again, if you need to back up and watch this again, please do so. Don't forget to supplement your understanding with watching a YouTube clip or two off of our website, and then follow up with the 10.2 reading. I hope that was helpful.